My name is Christine Dadafalza.
And my name is Jessica Agbeve.
For our enzyme webinar, we will be analyzing the WRN helicase.
So what is WRN helicase? Werner syndrome helicase, or WRN helicase, is an enzyme that belongs to the REC-Q DNA helicase family. REC-Q helicases travel on single-stranded DNA in a 3' to 5' direction and play essential roles in DNA replication and DNA repair. WRN specifically plays important roles in maintaining genomic stability, DNA repair, replication, transcription, and telomere maintenance. The WRN gene that encodes for the enzyme is highly conserved and can be found in many organisms such as bacteria, fungi, plants, humans, and other animals.
On this slide, we discuss the structure of WRN helicase. Using one of the bioinformatics websites called Swiss Model, we were able to examine its crystal structure, which is shown through this animation. WRN has 4 protein domains: the core helicase domain, the REC-Q terminal domain, the helicase and RNase D-like C-terminal domain, and the N-terminal 3' to 5' exonuclease domain.
In this chart, we listed some functions of each domain and its basic structures. The core helicase domain functions to unwind a variety of DNA substrates and is essential for hydrolysis of ATP. The overall structure of the core helicase domain consists of 2 REC-A-like helicase lobes, which are called D1 and D2. Each of them features a central 6-stranded parallel beta sheet on each side by helices and loops. The function of the REC-Q-C terminal domain is to mandate binding to the G quadruplex DNA and to stabilize binding to other DNA structures. The REC-Q-C terminal domain consists of 2 subdomains, the winged helix and zinc binding region. The zinc binding region has a single zinc ion tetrahedrally coordinated by 4 cysteine residues. It is also associated with the D2 domain. The winged helix domain is stretched away from the zinc and D2 domains and presents a reformed version of the normal winged helix fold with a longer wing too. The function of the helicase and RNase D-like terminal domain is to promote the localization of WRN helicase to specific areas where DNA is damaged. It also efficiently dissolves DNA intermediates known as holiday junctions. Lastly, the function of the N-terminal 3' to 5' exonuclease domain is to proofread bases after insertion and to degrade substrates that are likely to be found during DNA repair and as intermediates in DNA replication.
Moving on, we will review the functions of WRN helicase. This enzyme has both magnesium and ATP-dependent DNA helicase activity and 3' to 5' exonuclease activity towards double-stranded DNA with a 5' overhang. These activities allow it to maintain the structure and integrity of DNA. Examples would include double-stranded DNA repair, replication fork recovery, transcriptional regulation, telomere maintenance, DNA damage checkpoint, and early enhancer of RNA polymerase III.
In addition to its many functions, WRN helicase serves many roles in the replication fork. In Figure 1, WRN helicase indicated in blue unwinds the 5' flap and promotes strand displacement DNA synthesis and base excision repair. In addition, WRN helicase interacts with flap endonucleases, or FEN1s, indicated in green to promote a 5' flap endonuclease activity. Another role of WRN helicase in the replication fork is to unwind structures known as G quadruplexes that inhibit DNA synthesis. This can be seen in Figure B. And in Figure C, WRN helicase functions to unwind the lagging strand to initiate replication fork regression when the replication fork is stalled.
When talking about WRN helicase, it is important to note its connection to Werner syndrome, which is an autosomal recessive disorder characterized by accelerated aging. It is an inherited condition. The cause of Werner syndrome is a mutation in the WRN gene, which produces Werner syndrome enzymes. Symptoms of this disease would include wrinkles and loss of skin firmness, fat loss in upper layers of skin, hair graying, hair loss, distortion and discoloration of nails, and skin pigmentation issues. In addition, it is important to look at the health concerns for a person who has Werner syndrome. Health concerns would include skin ulcers, eye issues, dental issues, endocrine disorders, osteoporosis, hardening of the arteries, diabetes, and fertility issues. On top of that, there is an increased risk of cancer, which includes thyroid cancer, skin cancer, and sarcoma.
For our primary article, we chose to read an article titled Werner Syndrome Helicase Activity is Essential in Maintaining Fragile Site Stability.
Now, the main goal for this study was to determine whether WRN plays a role in the maintenance of fragile site stability. In this experiment, several methods were utilized to gain information about the WRN enzyme. It is known that WRN is primarily found in the nucleoli and that in response to DNA damage or replication fork arrest, it relocalizes to a nuclear foci. To verify this behavior, the researchers decided to test its response to partial inhibition of DNA replication. Wild-type fibroblasts were treated with 0.4 micromolar acetylcholine for 24 hours, pulse-labeled with 3 micrograms per milliliter bromodeoxyuridine for 30 minutes before fixation. Then, they were double immunostained with alpha WRN and alpha bromodeoxyuridine.
In order to test the hypothesis that the WRN plays a role in the maintenance of fragile site stability, the researchers decided to test the sensitivity of Werner syndrome cells to acetylcholine-induced replication slowdown, which required fragile site induction and preparation of cells. Acetylcholine is a reversible inhibitor of DNA replication that blocks the cell cycle at the S phase. Wild-type and Werner syndrome fibroblasts were exposed to various concentrations of acetylcholine, 0.05, 0.2, and 0.4 molar. Cell cultures were incubated with 0.2 micrograms per milliliter coalsamide solution at 37 degrees Celsius for 18 minutes. This was followed by numerous changes of fixated solution, a 3:1 methanol acetic acid. Cell suspension was dropped onto cold, wet slides and air dried overnight and kept at negative 20 degrees Celsius. After 24 hours, metaphase chromosomes were collected and assessed for the number of total gaps and breaks.
In addition, due to the possibility of a compensation for WRN deficiency in cells taken from Werner syndrome patients, researchers examined the effect of acetylcholine treatment in cells in which endogenous WRN was lowered. Human wild-type fibroblasts were transfected with control small interfering RNA. WRN was transfected with smart pool small interfering RNAs at the final concentration of 10 nanomolars. Transfection was performed using a high-perfect reagent. Western blotting probe with alpha WRN was used to test the reduction in the WRN protein levels. In order to do Western blotting, cells were washed with PBS and lysed in standard RIPA buffer. Cell lysates were resolved by SDS page and transferred to nitrocellulose. Incubation with antibodies was performed for 2 hours at room temperature. Proteins were visualized using ECL plus and normalized to the tubulin level in each extract.
To establish whether the increase in chromosomal gaps and breaks following acetylcholine treatment in Werner syndrome cells occurs at specific DNA locations, the researchers used FISH to examine the induction of most frequently expressed common fragile sites, FRA3b, FRA7H, and FRA16D. In wild-type and Werner syndrome fibroblasts, in order to perform the FISH test, bacterial artificial chromosome mapping to fragile or non-fragile site regions were used as probes. A mixture of the bacterial artificial chromosomes 94D19, 149J4, and 48E21 were used for FRA3D. Bacterial artificial chromosomes 36B6 were used for FRA7H, and bacterial artificial chromosomes 264L1 was used for FRA160. FISH signals were detected by incubation with anti-deoxygen rhodamine Fab fragments. Chromosomes were counterstained with DAPI. Hybridized metaphases were analyzed with epifluorescence microscopy. The researchers conducted the same experiment again in wild-type fibroblasts where endogenous WRN was regulated.
To determine whether one or both WRN enzymatic activities could affect fragile site stability, the researchers created Werner syndrome defective cell lines stably, expressing wild-type WRN or mutant versions of WRN, affecting either helicase or exonuclease activity. Then they utilize Western blotting to show the expression of WRN protein in cells stably expressing wild-type WRN or missense mutant forms of WRN with impaired exonuclease or helicase activity. In addition, hypersensitivity to camptothecin, a characteristic cellular phenotype of Werner syndrome cells, was tested. Cells were plated in a 6-well dish at a concentration of 3 times 10 to the power of 2 per well and treated with 1.5, 15, or 30 micromolar of camptothecin for 2 hours. Cell death was evaluated by counting cells using the trypan blue exclusion method. The trypan blue solution was diluted with an equal volume of PBS. Cells were detached and stained with 0.trypan blue stain solution. Blue cells were scored under a phased contrast optical microscope.
Now on this slide, we will be examining the most important figures from the experiment. In figure A, it shows the results of Western blotting. Levels of WRN protein expressed in Werner syndrome cells transfected with wild-type WRN complementary DNA and WRN lacking exonuclease or helicase activity were comparable to that of control cells. In figure B, it displays immunofluorescence staining of WRN protein. It exhibited the proper pattern of subnuclear localization, which was nucleoli under unperturbed conditions and diffused in the nucleoplasm after camptothecin-induced replication stress.
In figure C, hypersensitivity to camptothecin was tested and expression of wild-type WRN or missense mutant forms of WRN resulted in reduced cell death similar to the control cells. In figure D, expression of missense mutant forms of WRN protein in Werner syndrome cells resulted in a significant increase in chromosomal damage after acetylcholine exposure. In figure F, fish analyses performed on metaphases after 24 hours of treatment indicated that the induction of FRA3b, FRA7h, and FRA16d enhanced statistically only in Werner syndrome and WRNK577M cells. From this data, it can be concluded that the maintenance of common fragile site stability requires a WRN protein with intact helicase activity. In this experiment, there were a total of 4 separate tests that ultimately led up to our main goal in the final test. In the first test, it was proved that WRN is implicated in the response to replication slowdown induced by acetylcholine. In the second test, it demonstrated that Werner syndrome cells are sensitive to acetylcholine treatment and the loss of WRN is responsible for chromosome instability.
In the third test, it showed that WRN influences the stability of common fragile sites, both during normal DNA synthesis and in response to replication perturbation. In the last test, it indicated that the maintenance of common fragile site stability requires a WRN protein with intact helicase activity. And lastly, these are our references, and thank you for watching.